Dear students, today we are going to discuss about the cells of immune system. We will learn about various cell lineages such as the lymphoid cells and the myeloid cells which functions in our immune system. So, the immune response. The immune response to an antigen can be of two types: humoral or antibody mediated immunity and cellular or cell mediated immunity. The name humoral came from the word humor, which means body fluids. The humoral immunity is mediated by antibodies produced by B cells, which are present in blood and other body fluids. Hence the name humoral. The cellular immunity is mediated directly by cells. So it is known as cellular immunity. It is mediated through T lymphocytes and macrophages. The immune system is maintained by the lymphoreticular system. It is a complex organization of cells and it includes the lymphoid cells and the reticuloendothelial cells. Lymphoid cells. These cells are involved in specific immune response. They include lymphocytes. Lymphocytes mediate adaptive immune response. They mediate the hallmark attributes of immune response. They are the diversity, specificity, memory, and self and non-self recognition. So what are reticuloendothelial cells? They contribute to non-specific immunity. They include phagocytic cells. These cells play accessory roles in adaptive immunity. That is, they activate lymphocytes, carry out phagocytosis, secrete cytokines, etc., and they participate in inflammation. The origin of all these types of cells are the hematopoietic stem cell, which is present in the bone marrow. These stem cells are self-renewing and multipotent or pluripotent, that is, it is capable of differentiating into other cell types such as erythrocytes, granulocytes, monocyte, lymphocytes, etc. This process of differentiation is known as the hematopoiesis. During hematopoiesis, a hematopoietic stem cell differentiates along one of two pathways, either giving rise to a myeloid progenitor cell or a lymphoid progenitor cell. While myeloid progenitor cell generate progenitors of RBC, red blood cells, or erythrocytes, various white blood cells such as neutrophils, eosinophils, basophils, monocytes, mast cells, dendritic cells, etc., and platelets. A myeloid progenitor cell directly gives rise to a dendritic cell. There is a granulocyte monocyte progenitor coming from the myeloid progenitor cell, which will result in the formation of neutrophil or monocyte. A monocyte mature to become a macrophage. The eosinophil progenitor arising from the myeloid progenitor cell will become an eosinophil. A basophil progenitor cell will differentiate to form the basophil. The megakaryocyte will differentiate to form platelets, while the erythroid progenitor cell will differentiate to form the erythrocyte. While the common lymphoid progenitor cell, it will give rise to natural killer cells or null cells, T lymphocytes and B lymphocytes. And there are also some dendritic cells. The T cell progenitor cell arising from the lymphoid progenitor will mature or differentiate to become T helper cell or T cytotoxic cell, while the B cell progenitor will differentiate to become the B lymphocyte. In bone marrow, the differentiation of these stem cells into any type of cell depends on the presence and activity of several growth factors and cytokines. Colony stimulating factor, erythropoietin, etc. are few examples. Hematopoiesis occur with the help of a meshwork of stromal cells. And the stromal cells include fat cells, endothelial cells, fibroblasts, and few macrophages. All these stromal cells will provide a hematopoietic inducing microenvironment within the bone marrow which promote the growth and differentiation of various cell types. Coming to the lymphoid cells. During hematopoiesis in the bone marrow, these cells originate from the common lymphoid progenitor cells. These are lymphocytes, which are small round cells found in blood, lymph, lymphoid organs and in many other tissues. They constitute 20 to 40% of white blood cells or leukocytes in blood, and they are 99% of the cells in lymph. There are approximately 10 raised to 11 lymphocytes in the human body. According to their size, they are classified into three. They are small lymphocytes having 5 to 8 micrometer size, medium lymphocytes having 8 to 12 micrometer size, and large lymphocytes having 12 to 15 micrometer size. The small lymphocytes are the most numerous, and these are having slow motility. Depending on lifespan, the lymphocytes are classified into two: short-lived cells, also known as the effector cells, which survive for about 2 weeks, and long-lived or memory cells, which survive for 3 years to up to lifelong. Depending on the presence or absence of surface antigens or markers, also known as the cluster of differentiation CD markers, the lymphocytes are classified into three. We said the surface antigens or markers are also known as the cluster of differentiation CD markers, and there are about 150 CD markers identified so far. According to the presence or absence of these CD markers, the three classes of lymphocytes are B lymphocytes, T lymphocytes, and natural killer cells or null cells. So, the B lymphocyte. The name is derived from its site of maturation, that is bone marrow in mammals and bursa of Fabricius in birds. B cell has an extensive filamentous surface with numerous microvilli. They have membrane-bound immunoglobulin or antibody molecules on their surface which serve as receptors for antigen. Interaction between this membrane-bound antibody and the corresponding antigen induces the activation and differentiation of the B cell. Then it divides repeatedly and differentiates, generating a population of plasma cells and memory cells. Plasma cells have a very short lifespan of few days and many die in 1 or 2 weeks. Plasma cells synthesize and secrete antibody with the same antigen binding specificity as present in the original B cell. They can secrete more than 2000 molecules of antibody per second. The secreted antibodies may be one of the 5 classes of antibody, that is IgG or IgA or IgM or IgD or IgE. Memory B cells has a longer lifespan and they remains in the circulation for a long time, for up to years. They express the same membrane-bound antibody as the original parent B cell. What are the surface markers present in B cell? They are the surface immunoglobulin, CD45 and CD40, which functions in signal transduction, CD35 and CD21, which functions as a receptor for complement, CD32, which is a receptor for the immunoglobulin molecule, IgG, CD80 and CD86, which interact with the CD28 of T lymphocytes, and there are class 2 MHC molecules also. T lymphocytes, they derive their name from their site of maturation, thymus. These cells have T cell receptor or TCR, which are membrane receptors for antigen. These cells cannot bind to free antigen. They will bind to antigen presented along with particular molecules known as the major histocompatibility complex or MHC. In other words, to be recognized by T cells, antigens must be displayed together with MHC molecules on the surface of certain cells known as antigen presenting cells. The common antigen presenting cells are B cells, macrophages and dendritic cells, etc. There are T helper cells or Th cells and T cytotoxic cells or Tc cells. Th cell express the CD marker CD4 and Tc cell express CD8 glycoprotein on their surface. The Th cells or the CD4 T lymphocytes get activated when it encounters an antigen complex with MHC class II molecule by an antigen presenting cell such as B cell, macrophage or dendritic cell. The activated Th cell will secrete various growth factors known as cytokines, which will further activate B cells, T cytotoxic cells or Tc cells and a variety of other cells that participate in an immune response. There are two types of T helper cell response. They are Th1 response and Th2 response. Th1 cell response support inflammation and activates certain T cells and macrophages, whereas the Th2 response activates B cells and immune responses which depend upon the presence and activity of antibodies. The T cytotoxic cells or Tc cells, which have a CD8 marker on their surface, they will get activated when they interact with an antigen complex with MHC class I molecule. These are presented on altered cells such as virus infected cells, cancer cells, graft cells, etc. Activation of Tc cell occurs, which results in the proliferation and differentiation of the Tc cell to become the cytotoxic T lymphocytes or CTL. The cytotoxic T lymphocyte recognize and eliminate the altered self cells such as the virus infected cells, cancer cells or graft cells. And these cells, these cytotoxic T lymphocytes are generally responsible for killing the virus infected cells, transplanted cells and cancer cells. Another subpopulation of T lymphocytes is known as the T suppressor cells, and they help to suppress the humoral and the cell mediated immune system. The common surface markers of T lymphocytes are T cell receptor or TCR, thymus specific antigens, CD2 and CD3 which functions in signal transduction, CD4 or CD8 where CD4 binds to class II MHC molecules and CD8 binds to class I MHC molecule, CD28 which will bind to the B7 receptor on the surface of antigen presenting cells, CD45 which functions in signal transduction, and there is a CD40 ligand. Next, natural killer cells or NK cells or large granular lymphocytes or LGL. These are large granular lymphocytes that do not express surface markers typical of either T lymphocyte or B lymphocyte. They possess cytotoxicity against a wide range of tumor cells. These cells are not antibody dependent or MHC restricted. They lack immunological specificity and memory. Natural killer cells recognize the potential target cells either through the unusual profile of surface antigens on the surface of target cell or the CD16 marker which is present on the surface of natural killer cell will bind to the antibodies which are bound on the surface of the target cells. The process where the CD16 marker of the natural killer cell will bind onto the antibody present on the surface of the target cell and the subsequent destruction of the target cell is known as antibody dependent cell mediated cytotoxicity. Once bound to the target cell, the natural killer cell will release cytolytic factors such as perforin which will cause the formation of transmembrane pores. Through these transmembrane pores, the cytotoxic factors such as tumor necrotic factor beta or TNF beta will enter the cell and will kill the cell through apoptosis. Lymphokine activator killer cells or LAK cells are natural killer cells treated with interleukin 2, a cytokine, and these cells, the LAK cells, are cytotoxic towards tumor cells. The mononuclear phagocytes consist of monocytes, which circulate in blood, and macrophages, which are present in tissues. During hematopoiesis, they originate from the myeloid progenitor cell, which will differentiate into granulocyte monocyte progenitor cells. The granulocyte monocyte progenitor cells will differentiate into promonocytes, and the promonocytes then leave the bone marrow and enter the blood, where they differentiate into monocytes. Monocytes then circulate in the bloodstream, and during this circulation they will enlarge and then migrate into the tissues, and in the tissues they will become macrophages. Differentiation of a monocyte into a tissue macrophage involves the following stages. The cell will become 5 to 10-fold large. The number and complexity of intracellular organelles will increase. The cells will acquire increased phagocytic ability. They will produce higher levels of hydrolytic enzymes, and the cells will begin to secrete a number of soluble factors. Monocytes are the largest of the lymphoid cells which are present in the peripheral blood. Their size is approximately 12 to 15 micrometer, and they have a half-life of about 3 days. Tissue macrophages are larger, and they have a size of 15 to 20 micrometer. The macrophages are dispersed throughout the body, and they can survive for months. Macrophages are either free macrophages, which travel throughout the tissues through amoeboid movement, and these are known as free or wandering macrophages. Or the macrophages may reside in a particular tissue. They cannot move, they cannot travel, and they are known as the fixed macrophages. Macrophages are named according to their tissue location. For example, alveolar macrophages are present in lungs, histiocytes are present in connective tissue, Kupffer cells are present in liver, mesangial cells are the macrophages present in kidney, microglial cells are the macrophages present in brain, and osteoclasts are the macrophages present in bone. Functions of macrophages include phagocytosis, antigen presentation, and secretion of cytokines. The first function is phagocytosis. Macrophages are capable of ingesting and digesting exogenous antigens such as microorganisms or other insoluble particles or antigens, and they can also ingest and digest endogenous matter such as injured or dead host cells, cellular debris, or activated clotting factors, etc. The process of this ingestion and digestion is known as phagocytosis. The second function is antigen presentation. During antigen presentation, the phagocytosed antigen is digested within the macrophage into peptides. These peptides will associate with class II MHC molecules, and the peptide class II MHC molecule complex will move to and display at the macrophage surface, membrane surface. This processing and presentation of antigen is essential to activate T helper cells, which is further essential for the development of humoral and cell-mediated immune responses. The third function is the secretion of cytokines. A number of important cytokines such as interleukin 1, tumor necrotic factor alpha, and interleukin 6 that promote inflammatory responses are secreted by activated macrophages. So how the macrophage activity is induced or enhanced? The macrophage activity can be enhanced by the cytokines secreted by activated T helper cells, by mediators of the inflammatory response, and by components of bacterial cell walls. One of the most potent activators of macrophage is interferon gamma, secreted by the activated T helper cells. Phagocytosis by macrophage can be enhanced by antibodies. The macrophage membrane has receptors for antibody and they can bind to cells or antigens bound to the antibody. So, the antibody functions as an opsonin, that is a molecule which binds to both antigen and macrophage, which act as a bridge between the antigen and macrophage, and such antibody molecules can enhance phagocytosis, the process by which antigens are rendered more susceptible to phagocytosis by this type of antibody molecules is known as opsonization. Coming to the granulocytic cells, these cells are originated from myeloid progenitor cells. The granulocytes are classified as neutrophils, eosinophils or basophils or mast cells on the basis of cellular morphology and cytoplasmic staining characteristics. The neutrophils are 50 to 70% of the circulating white blood cells or WBC. They have a multi-lobed nucleus and a granulated cytoplasm that stains with both acid and basic dyes. They are also known as polymorphonuclear leukocytes due to their multi-lobed nucleus. The second one, the eosinophils, they are 1 to 3% of circulating white blood cells. They have a bilobed nucleus and a granulated cytoplasm that stains with the acid dye eosin red. The third type is the basophils. They are less than 1% of the circulating white blood cells and they have a lobed nucleus and a heavily granulated cytoplasm that stains with the basic dye methylene blue. Both neutrophils and eosinophils are phagocytic, whereas the basophils are not phagocytic. The neutrophils have a lifespan of only few days. Neutrophils are active phagocytic cells and they are the cells which arrive first at the site of inflammation. Movement of the circulating neutrophils from blood into the tissues is known as extravasation. The neutrophils contain primary and secondary granules. The larger and denser primary granules contain peroxidase, lysozyme and various other hydrolytic enzymes, while the smaller secondary granules contain collagenase, lactoferrin and lysozyme. Eosinophils, they are motile phagocytic cells and they play a role in the defense against parasitic organisms. Basophils are non-phagocytic granulocytes. They contain pharmacologically active substances such as histamine, serotonin, etc. in their cytoplasmic granules, which play a major role in certain allergic responses. The mast cells, they are found in tissues. They are not present in circulation. They are found in tissues including skin, connective tissues of various organs and mucosal epithelial tissue of the respiratory, genitourinary and digestive tracts. Like basophils, mast cells have cytoplasmic granules that contain pharmacologically active substances. The mast cells and basophils play an important role in the development of allergies. Now, the dendritic cells. They originate from myeloid progenitor cells and also from lymphoid progenitor cells. The dendritic cell is named so because it is covered with long membranous extensions that resemble the dendrites of nerve cells. 4 types of dendritic cells are known. They are Langerhans cell, interstitial dendritic cells, myeloid dendritic cells and lymphoid dendritic cells. The function of dendritic cells is the presentation of antigen to T helper cells. Dendritic cells are the more potent antigen presenting cells than macrophages and B lymphocytes. Immature or precursor dendritic cells acquire antigen by phagocytosis or endocytosis. Then the antigen will be processed and it will be presented along with class II MHC molecules to the T helper cells. So, we were discussing about cells of immune system, and we discussed T lymphocytes, natural killer cells, B lymphocytes, macrophages, neutrophil, eosinophil, basophil, mast cells and dendritic cells. So the B lymphocyte and T lymphocyte and the natural killer cells, they originate from the lymphoid progenitor cell. There are also few dendritic cells coming from the lymphoid progenitor cells. And we discussed about neutrophil, monocyte, macrophage, eosinophil, basophil, etc. which originate from the myeloid progenitor cells. A few dendritic cells also originate from the myeloid progenitor cells. All these are the cells of immune system. Thank you so much for listening.